So here's something to think about as you settle in tonight. Right now, at this very moment, there are roughly 86 billion neurons inside your skull. 86 billion tiny living cells, each one reaching out and touching thousands of others, forming something like 100 trillion connections. And those connections are firing right now, as you lie here, producing little bursts of electricity that ripple across your brain in waves. Some of them are keeping your heart beating. Some are processing the sound of my voice. Some are holding on to a thought you had earlier today, a conversation, a worry, a half-remembered song. And some of them, right now, are doing something almost absurdly strange. They are trying to understand themselves. Your brain is listening to a story about brains. The organ that makes you you is attempting to comprehend its own machinery. And if that doesn't strike you as one of the most remarkable things in all of nature, just sit with it for a second, because it really is. And that's what we're going to talk about tonight. Animal brains, how they work, how they evolved, how we figured out what they do. We're going to start way, way back, before anyone had the faintest clue what the brain was for, and we're going to walk all the way through to the astonishing things neuroscientists know today, and we'll take our time with it. There's no rush at all. If you drift off somewhere along the way, that's perfectly fine. Your brain will keep right on doing its thing, whether you're awake to appreciate it or not. That's sort of the whole point. Let's start with a question that might seem obvious now, but was genuinely confusing for a very, very long time. What does the brain actually do? What is it for? Because for most of human history, people had no idea. And some of the smartest people who ever lived got it completely, spectacularly wrong. Take the ancient Egyptians. They were brilliant engineers, extraordinary architects, masters of medicine in many ways. And when they mummified their dead, they took extraordinary care to preserve the organs they believed the person would need in the afterlife. The heart was sacred. They left it in the body. The lungs, the liver, the stomach, all carefully preserved in canopic jars. But the brain? They pulled it out through the nose with a hook and threw it in the garbage. They didn't think it did anything important, and they weren't alone. Aristotle, one of the greatest thinkers in all of human history, believed the brain was a cooling system for the blood. That's it, a radiator. He noticed that the brain was moist and cool compared to the heart, which was warm and active and seemed connected to emotions, and he concluded that the heart was the seat of intelligence while the brain just cooled things down. He was wrong, but he was wrong for interesting reasons. Not everyone agreed with Aristotle, even in the ancient world. Hippocrates, the Greek physician, had argued forcefully that the brain was the seat of thought, emotion, and sensation, and the physician Galen, who lived in the 2nd century in Rome and served as doctor to the gladiators, pushed this further through actual dissection. Galen showed that cutting certain nerves caused paralysis in specific parts of the body. He demonstrated that the brain, not the heart, controlled voluntary movement. He even identified the difference between sensory nerves, which carry information into the brain, and motor nerves, which carry commands outward. That's a fundamental distinction, and Galen figured it out nearly 2,000 years ago. But Galen also got a lot wrong. He believed the important part of the brain wasn't the solid tissue itself, but the fluid-filled cavities inside it, the ventricles. He thought mental functions happened through the movement of fluid, not through the brain tissue. This ventricular doctrine became dogma for over a millennium. Scholars drew elaborate diagrams showing how different mental faculties lived in different ventricles: perception in the front, reason in the middle, memory in the back. It was neat, tidy, and almost entirely wrong. 14 centuries of people looking at the brain and focusing on the wrong part of it. The shift started slowly in the Renaissance. Vesalius challenged Galen's anatomy. Thomas Willis, working in Oxford in the 1660s, was the first to seriously argue that mental functions were located in the brain's solid substance, not its fluid. The circle of Willis at the base of the brain is named after him. He tried to assign specific mental functions to specific brain regions, and the basic framework, that different parts of the brain do different things, was a genuine breakthrough. But here's the thing. Even as anatomists were getting better at describing what the brain looked like, they were still completely in the dark about how it actually worked. They could draw it, map its blood vessels, identify its major structures. But what was the brain doing, physically, when you had a thought, when you moved your hand, when you felt pain? What was the actual mechanism? That question would take another 200 years to answer, and the answer, when it came, would involve a whole new way of thinking about the body. It would involve electricity. And this brings us to one of the most dramatic experiments in the history of science, and one of the most colorful characters. His name was Luigi Galvani, and the year was 1780, and the place was Bologna, Italy. Galvani was a physician and physicist, and the story, which may be somewhat embellished, but the core is real, goes like this. Galvani was in his laboratory, dissecting frogs, as anatomists of the period often did. A dead frog was lying on a table near an electrostatic machine, one of those devices that generates sparks of static electricity, and Galvani's assistant touched a metal scalpel to a nerve in the frog's leg. At the exact moment, a spark discharged from the machine. The frog's leg twitched. The dead frog's leg kicked, as if it were alive. Now, this was electrifying, in every sense of the word. Galvani repeated the experiment, varied it, tested it dozens of ways. He found he could make frog legs twitch by touching different metals to the nerve, even without the static machine. He hung frog legs from iron railings with brass hooks and watched them kick during thunderstorms. And he concluded that there was an innate electrical force within living tissue itself, an animal electricity, he called it, that was fundamentally different from the electricity in machines and lightning. The body, Galvani believed, generated its own electricity, and this electricity was what made muscles move and nerves conduct. He was mostly right. His contemporary Alessandro Volta argued, correctly, that some of Galvani's effects were produced by the contact between different metals, and this debate led Volta to invent the electric battery. But Galvani's core insight, that nerves transmit signals using electricity, was one of the most important discoveries in the history of biology. If the brain ran on electricity, maybe you could study it the way physicists studied circuits, and people immediately tried. In the decades after Galvani, researchers began stimulating animal brains with electrical current. In 1802, Galvani's nephew Giovanni Aldini gave a public demonstration in London applying current to the head of a recently executed criminal. The corpse's jaw clenched. One eye opened. Mary Shelley was almost certainly influenced by these demonstrations when she wrote Frankenstein in 1818. But the really important work was quieter. In the 1870s, two German researchers, Gustav Fritsch and Eduard Hitzig, applied small electrical currents to the exposed brains of dogs, reportedly performing their early experiments on Fritsch's wife's dressing table because they didn't have proper laboratory space. They showed that stimulating specific spots on the cerebral cortex caused specific muscles on the opposite side of the body to move. Touch one spot, the front leg twitches. Move a few millimeters, the hind leg moves instead. The brain had a map of the body laid out across its surface. This was the beginning of what neuroscientists call localization of function, the idea that specific mental and physical functions are handled by specific brain regions. and it would lead, within a few decades, to the discovery that the brain is organized in ways that are both extraordinarily precise and deeply strange. There's a strip of cortex, for instance, called the motor cortex, where each body part is represented in a specific location, but the sizes of the representations aren't proportional to the actual sizes of the body parts. Your hands and your lips and your tongue have enormous territories on the motor cortex, far larger than what's devoted to your back or your legs, because hands and lips and tongue require incredibly fine motor control. If you built a human figure, with body parts sized according to how much motor cortex they occupy, you'd get a grotesque little creature with enormous hands, huge lips, and a tiny torso. That figure has a name, the motor homunculus, and it's one of the most memorable images in neuroscience. There's a matching sensory homunculus, too, for the somatosensory cortex, where touch and pressure are processed. Same distortion. Your fingertips and your lips take up a huge proportion of the sensory map, because those are the parts of your body that have the most nerve endings, the finest discrimination. But we're getting ahead of ourselves a little. Because while the localization story was developing on one front, an entirely different revolution was happening at a much smaller scale, down at the level of individual cells. And this revolution would answer a question that had been nagging at scientists for decades. What, physically, is the brain made of? What are the basic building blocks? And the answer turned out to be stranger and more beautiful than anyone expected. The story of how we found it involves two stubborn scientists, a bitter rivalry, a shared Nobel Prize, and a staining technique involving silver and potassium dichromate that would reveal the hidden architecture of the mind for the first time in human history. And it starts in Italy, in a hospital kitchen, in the early 1870s, with a young doctor named Camillo Golgi, who was about to make a discovery that would change everything, even though he would spend the rest of his life misunderstanding what he had found. Golgi was working at a hospital in Abbiategrasso, a small town near Milan. He didn't have a proper laboratory. He didn't have funding. He was performing his experiments in a converted kitchen, by candlelight, in whatever spare time he could scrape together between his clinical duties. And he was trying to do something that had frustrated anatomists for decades. He was trying to see the structure of brain cells. Here's the problem he was up against. Brain tissue, under a microscope, looks like an unreadable mess, a tangle. Imagine looking at a bowl of spaghetti where every strand is the same color as every other strand, and they're all interwoven so tightly you can't tell where one ends and another begins. That's roughly what brain tissue looks like when you slice it thin and put it under a lens. Everything runs into everything else. The cells are densely packed. Their extensions are intertwined. And with the staining techniques available at the time, you could see individual cell bodies as small dots, but the branches extending from them, the thin, thread-like projections that are absolutely crucial to how the brain works, those were invisible. You couldn't trace them. You couldn't see where they went. You couldn't tell whether they connected to other cells or not. The brain's wiring was hidden. And then Golgi stumbled onto something remarkable. In 1873, he developed a staining method that used silver chromate, a combination of potassium dichromate and silver nitrate. He soaked brain tissue in this solution, and something almost magical happened. For reasons that aren't entirely understood even today, the stain doesn't label every cell. It labels only a small random fraction of them, maybe 1 or 2%. But the cells it does label, it labels completely. Every branch, every tiny fiber, every delicate extension, the entire cell lights up in stark black against a clear yellow background, like a tree silhouetted against a winter sky. Suddenly, For the first time in history, you could see what a brain cell actually looked like. And the shapes were extraordinary. These weren't simple round blobs. They were elaborate, branching structures of staggering complexity. From each cell body, thick branches extended outward and then subdivided into finer and finer branches, reaching in all directions. And from one side of the cell, a single long fiber, much thinner than the branches, extended outward, sometimes for enormous distances, before itself branching at its far end. These cells were nothing like anything else in the body. They were built for reaching, for connecting, for touching other cells across vast distances. Golgi had invented what is now called the Golgi stain, and it remains one of the most important technical innovations in the history of neuroscience. Without it, we simply could not have understood the brain's structure. But here's the irony, and this is one of those stories that shows how even brilliant scientists can see the evidence and draw the wrong conclusion. Golgi looked at his beautifully stained brain tissue and concluded that the branches of different cells fused together into one continuous network, like a fishing net. He believed the brain was not made of separate individual cells, but was instead a single interconnected mesh of tissue, a reticular network where everything was physically joined to everything else. Signals, in his view, flowed through this continuous web, the way water flows through pipes. Individual cells weren't the fundamental units. The network was. This was called the reticular theory, and it was reasonable, given what you could see. The branches of brain cells do come very close to each other. They do seem to overlap and interweave. And with the technology of the 1870s, it was genuinely hard to tell whether they were fusing or merely touching. Golgi committed to the reticular theory and defended it with increasing stubbornness for the rest of his career. And now the other half of the story walks in. Santiago Ramon y Cajal. If Golgi is one of the most important figures in neuroscience history, Cajal is the most important. Period. Full stop. There's a reasonable argument that he is the single most influential neuroscientist who ever lived, and his story is one of those tales of sheer determination and obsessive genius that almost defies belief. Cajal was born in 1852 in Petilla de Aragon, a tiny village in the mountains of Navarre in northern Spain. His father was a barber surgeon, not a doctor in the modern sense, but a practical medical man who also cut hair and performed minor procedures. The family was poor. Young Santiago was, by all accounts, a handful. He was headstrong, artistic, constantly getting into trouble. He wanted to be a painter. His father wanted him to be a doctor. The compromise, if you can call it that, was medicine, which his father essentially forced on him. Cajal would later say that his artistic training was the most important preparation for his scientific career, because what he would ultimately do, better than anyone in history, was draw. He drew what he saw through the microscope with an accuracy, a beauty, and an insight that have never been surpassed. His drawings of neurons are still used in textbooks today. They are works of art and works of science simultaneously. Cajal encountered Golgi's staining method in 1887, more than a decade after Golgi had invented it. He was 35 years old, working at the University of Barcelona, largely unknown outside Spain. A colleague showed him some brain tissue stained with the Golgi method, and Cajal was thunderstruck. He later described the moment with almost religious intensity. He saw those individual cells lit up against the clear background, and he knew instantly that this technique was the key to unlocking the brain's secrets. He threw himself into the work with manic energy. He modified the Golgi stain, improved it, adapted it for different tissues. He worked obsessively, spending 16 hours a day at his microscope. His wife, Silveria, essentially ran the household and raised their children, while Cajal lived inside his microscope. He examined brain tissue from every source he could find: birds, frogs, mice, rabbits, humans, and gradually, painstakingly, he built up a picture of the brain's architecture that was completely different from Golgi's. Where Golgi saw fusion, Cajal saw separation. Where Golgi saw a continuous mesh, Cajal saw individual cells, each one distinct and independent, reaching toward other cells but never quite fusing with them. The branches came close, agonizingly close, but there was always a tiny gap. The cells were separate entities. They communicated across that gap, but they didn't merge. The brain was not a single continuous network. It was a population of individual cells, each one a discrete unit, each one receiving signals from some neighbors and sending signals to others. This was the neuron doctrine, and it was one of the most consequential ideas in all of biology. The brain is made of individual cells, neurons, and those neurons are the fundamental structural and functional units of the nervous system. Everything the brain does, every thought, every sensation, every memory, every movement, every emotion, is produced by the activity of individual neurons communicating with each other across tiny gaps. Cajal couldn't see those gaps directly. The technology wasn't good enough, but he inferred their existence from the way the cells were shaped and arranged. He was right. The gaps would later be confirmed by electron microscopy in the 1950s, and they would get a name that has become one of the most important words in neuroscience. They're called synapses, a term coined by the English physiologist Charles Sherrington in 1897 from the Greek word for clasp or junction. In 1906, the Nobel Prize in Physiology or Medicine was awarded jointly to Golgi and Cajal, and the ceremony itself was one of the most awkward events in Nobel history, because Golgi, in his acceptance speech, stood up and vigorously argued for his reticular theory, essentially contradicting the very evidence that Cajal had spent 20 years amassing. And then Cajal stood up and, with barely contained fury, dressed in diplomatic language, presented the neuron doctrine. Two men sharing the highest honor in science, and they fundamentally disagreed about the most basic question in their field. History has sided overwhelmingly with Cajal. The neuron doctrine is as well established as any fact in biology. Golgi's reticular theory is wrong. But it took extraordinary work, extraordinary evidence, and extraordinary stubbornness to settle the matter. Now, here's where things get really interesting. Because once you accept that the brain is made of individual neurons, the next question is obvious. How does a neuron actually work? What is it doing, physically, when it transmits a signal? And to understand that, we need to talk about what a neuron looks like and how it's built, because the architecture of the cell is intimately tied to its function. Imagine you're looking at a single neuron through Cajal's microscope. What you see is something that looks roughly like a tree. At the center is the cell body, or soma, a roundish blob containing the nucleus, the DNA, the protein-making machinery, all the standard cellular equipment. Extending outward from the cell body is a network of thick branches that subdivide into thinner and thinner branches, reaching out in all directions. These are called dendrites, from the Greek word for tree. The dendrites are the cell's input devices. They receive signals from other neurons, and then, extending from the cell body in a different direction, is a single, long, thin fiber called the axon. The axon is the cell's output cable. It can be short, a fraction of a millimeter in the brain, or it can be astonishingly long. The axon that runs from the base of your spinal cord to the tip of your big toe is about a meter long, a single unbroken fiber, one cell stretching the entire length of your leg. In a giraffe, those axons can be several meters long. One cell, one fiber, several meters. At the far end, the axon branches into many fine terminals, and each terminal comes very close to the dendrite, or cell body of another neuron. That near contact point is the synapse, and there are a lot of them. A single neuron in the cerebral cortex typically receives input at something like 7,000 synapses. Some neurons receive more than 100,000. When you multiply that by 86 billion neurons, you get the roughly 100 trillion synaptic connections in the human brain. That number is larger than the number of stars in the Milky Way galaxy. And every one of those connections is doing something. Every one of them is transmitting or withholding a signal, strengthening or weakening, adjusting the brain's circuitry moment by moment. But what's actually traveling down the axon? What's the signal? We said earlier that the nervous system runs on electricity, and that's true. But it's a very different kind of electricity from what flows through the wires in your wall. The electricity in a copper wire is carried by free electrons zipping along the metal at enormous speeds. The electricity in a neuron is carried by ions, charged atoms, mainly sodium, potassium, calcium, and chloride, flowing through tiny channels in the cell membrane. It's slower, it's wetter, and the mechanism is ingeniously different. Here's how it works. And this is one of the most beautiful pieces of biology ever discovered. When a neuron is resting, just sitting there not doing anything, its inside is negatively charged relative to the outside. There's a voltage difference across the cell membrane of about -70 millivolts. That's not much, about 1/20th of the voltage in a AA battery, but it's critical. The neuron maintains this resting potential by actively pumping ions back and forth across its membrane, using molecular pumps that require energy. Sodium ions are pumped out, potassium ions are pumped in. This establishes an uneven distribution, more sodium outside, more potassium inside, and because the membrane is slightly more permeable to potassium than to sodium at rest, a net negative charge builds up inside the cell. The neuron is like a tiny battery, charged and waiting. Now, when a signal arrives at the dendrites, it can change that voltage. Incoming signals from other neurons can either push the voltage closer to zero, which is called depolarization, or push it further from zero, which is called hyperpolarization. Depolarization makes the neuron more likely to fire. Hyperpolarization makes it less likely. The neuron is essentially integrating thousands of tiny pushes and pulls from all its incoming synapses, moment by moment, doing a kind of unconscious arithmetic. And if the sum of all those inputs pushes the voltage at the base of the axon past a critical threshold, roughly -55 millivolts, something dramatic happens. The neuron fires. And fires is the right word, because what happens next is explosive and all or nothing. Voltage-sensitive sodium channels in the membrane snap open. Sodium ions flood into the cell, driven by both the concentration gradient and the electrical gradient. The voltage at that spot shoots up from -70 millivolts to about +40 millivolts in less than a millisecond. That's a swing of over 100 millivolts. And that voltage spike triggers the sodium channels in the next stretch of membrane to open, which triggers the next stretch, which triggers the next, and the signal propagates down the axon like a wave, like a line of dominoes falling. This is the action potential, and it is the fundamental unit of communication in the nervous system. Every thought you've ever had, every movement you've ever made, every sensation you've ever felt was carried, at some point, by action potentials propagating along axons. And here's what's elegant about it. The action potential is always the same size. It doesn't matter how strong the stimulus was. Once the threshold is crossed, the signal is full strength. It's digital, in a sense. On or off, fire or don't fire. Neurons don't send stronger signals by sending bigger action potentials. They send them by firing more frequently. More action potentials per second means a stronger signal. It's like Morse code, where the information is in the pattern and timing of the pulses, not in the loudness of each individual beep. Now, the people who figured out exactly how action potentials work, who measured the currents, identified the ion channels, and wrote the mathematical equations that describe the whole process. Those people are Alan Hodgkin and Andrew Huxley, working at the University of Cambridge in the late 1940s and early 1950s. And the way they did it is one of the great stories of experimental biology, involving ingenuity, patience, and a very unusual piece of biology. The problem was that neurons are tiny. Most axons are a micrometer or less in diameter. In the 1940s, there was no technology that could get an electrode inside something that small. But nature provided a loophole. The Atlantic squid has a giant axon, about 1 millimeter in diameter, roughly 1,000 times thicker than a typical mammalian axon. The squid uses it for its escape jet because bigger axons conduct faster, and for an escape response, speed is life. For Hodgkin and Huxley, it was a gift. They could actually insert a glass electrode inside it and measure voltage changes directly. Working at the Marine Biological Association laboratory in Plymouth, England, they spent summers dissecting squid and recording nerve impulses. The work was interrupted by World War II, but between 1948 and 1952, they produced a series of papers considered among the most important in the history of biology. Using a technique called the voltage clamp, they identified 2 separate ion currents, a fast inward sodium current that produces the rapid upstroke of the action potential, and a slower outward potassium current that brings the voltage back down. They wrote mathematical equations describing the whole process, and when they ran the equations through a room-sized mechanical calculator, the computed action potential matched the real one almost perfectly. For the first time, nerve impulse conduction was understood at the level of physics and chemistry. No mysterious vital force, just ions flowing through channels in a membrane. Hodgkin and Huxley won the Nobel Prize in 1963, and their model remains the foundation of computational neuroscience to this day. But we've talked about what travels along the axon. What happens when the signal reaches the end? What happens at the synapse, at that tiny gap between one neuron and the next? And this brings us to another beautiful story, one that involves a dream, a pair of frog hearts, and one of the most elegant experiments in the history of science. The man's name was Otto Loewi, and he was an Austrian pharmacologist working in Graz in the early 1920s. And the question he was wrestling with was the same one everyone in neuroscience was arguing about. How do signals cross the synapse? There were 2 camps. One camp thought the signal was electrical, that the action potential in one neuron somehow jumped directly across the gap to the next neuron, like a spark jumping between 2 wires. This was called the electrical hypothesis, and it had the appealing simplicity of keeping everything in the same currency. Electricity in, electricity across, electricity out. The other camp thought the signal was chemical. They believed that when an action potential reached the end of an axon, it triggered the release of a chemical substance, and that chemical drifted across the gap and stimulated the next neuron. This was called the chemical hypothesis, and it was trickier because it required a whole additional mechanism. You had to have a chemical stored in the axon terminal, a mechanism to release it, a way for the chemical to cross the gap, and a receptor on the other side to detect it. It was more complicated, And in science, the simpler explanation usually wins, unless the evidence forces you to accept the complicated one. Lowy had been thinking about this for years. He suspected the chemical hypothesis was right, but he couldn't figure out how to test it. And then, on the night before Easter Sunday in 1921, he woke up in the middle of the night with an idea for an experiment. He scribbled it down on a piece of paper and went back to sleep. When he woke up in the morning, he looked at his notes and couldn't read them, couldn't make out his own handwriting. He spent the entire day trying to remember what the idea was, and he couldn't. It was gone. And then, the next night, at 3 o'clock in the morning, the idea came back. This time, he didn't bother writing it down. He got dressed, went straight to his laboratory, and performed the experiment on the spot. Here's what he did, and it's beautiful in its simplicity. He took 2 frog hearts. In frogs, the heart is controlled by a nerve called the vagus nerve, and when you stimulate the vagus nerve, the heart slows down. Lowy set up the 2 hearts so that the fluid bathing the first heart could flow onto the second heart. Then he stimulated the vagus nerve of the first heart. As expected, the first heart slowed down. And then, here's the crucial moment. The fluid that had been bathing the first heart flowed onto the second heart, and the second heart slowed down too, even though the second heart's vagus nerve hadn't been stimulated. Even though there was no direct electrical connection between the 2 hearts, the implication was unmistakable. When the vagus nerve fired, it released a chemical substance into the fluid around the first heart, and that chemical substance was enough to slow down the second heart when the fluid reached it. Synaptic transmission was chemical, not electrical. The nerve released a substance. Lowy initially called it Vagusstoff, Vegas stuff in German. We now know it as acetylcholine, and it was the first neurotransmitter ever identified. Think about what that means for a moment. Every time one neuron talks to another, every time you have a thought, form a memory, feel an itch, move a finger, what's actually happening at the synapse is this. An action potential arrives at the axon terminal, triggers tiny packets of chemical called vesicles to fuse with the cell membrane, dumping their chemical payload into the synaptic gap. And those chemical molecules drift across the gap in a fraction of a millisecond and bind to receptor proteins on the receiving neuron, which then respond by opening ion channels and either exciting or inhibiting that neuron. It's a molecular handshake, happening 1 trillion times a second across your brain. And here's what's really clever about chemical synaptic transmission. Because the signal has to go through this chemical middleman, the synapse becomes a point of control, a point where the signal can be modified, amplified, weakened, or blocked entirely. If transmission were purely electrical, like a wire, the signal would just pass through unchanged. But because it's chemical, you can adjust the amount of neurotransmitter released. You can change how many receptors are on the receiving side. You can have enzymes that break down the neurotransmitter faster or slower. You can have reuptake pumps that suck the neurotransmitter back up before it can act. All of these are knobs and dials that the brain can adjust, and adjusting them is a huge part of how the brain learns and adapts. We'll come back to that later. Now, acetylcholine was just the beginning. Over the following decades, neuroscientists discovered that the brain uses dozens of different chemical neurotransmitters, each with its own personality, if you will. There's dopamine, which plays a central role in motivation, reward, and movement. It's the neurotransmitter that's depleted in Parkinson's disease, which is why people with Parkinson's have difficulty initiating movements. There's serotonin, which is involved in mood, sleep, appetite, and a whole host of other things. Most modern antidepressants work by increasing serotonin levels at the synapse. There's norepinephrine, involved in alertness and the stress response. There's GABA, which stands for gamma-aminobutyric acid, and it's the brain's main inhibitory neurotransmitter. It tells neurons to calm down, to not fire. Without GABA, neurons would fire uncontrollably, which is essentially what happens during a seizure. And there's glutamate, the brain's main excitatory neurotransmitter, the one that says, go. Together, glutamate and GABA account for the vast majority of synaptic transmission in the brain, one pushing the accelerator and the other pressing the brake. And this is where we can understand something really practical. A huge number of drugs that affect your mind work by interfering with neurotransmitter systems. Caffeine blocks receptors for adenosine, a neurotransmitter that promotes sleepiness. Alcohol enhances GABA, the inhibitory neurotransmitter, which is why it slows your thinking and makes you drowsy. Cocaine blocks the reuptake of dopamine, leaving more of it in the synapse, which produces intense euphoria by hijacking the brain's natural reward system. Neurotransmitters are the brain's chemical vocabulary, and their pharmacology is the basis of psychiatric medications, anesthetics, painkillers, and much more. Lowy's dream experiment opened the door to an entire medical discipline. Lowy won the Nobel Prize in 1936, shared with Henry Dale, who had done crucial work identifying acetylcholine and its mechanisms. But Lowy's story has a darker chapter. He was Jewish, living in Austria, and when the Nazis annexed Austria in 1938, they arrested him. They forced him to transfer his Nobel Prize money to a Nazi-controlled bank. He was eventually allowed to emigrate, and he spent the rest of his career in New York, at New York University, where he continued working until his death in 1961. The experiment that came to him in a dream ended up saving his life. It gave him the international reputation and the connections that helped him escape. Science is woven into history in ways that are sometimes hard to separate. Now, let's shift gears a little. We've been talking about how individual neurons work and how they talk to each other. But the brain doesn't do anything useful with single neurons. Everything interesting happens when neurons work together in circuits and systems. And one of the most fascinating places to see this is in how the brain processes sensory information. How do you see? How do you hear? How do you feel the weight of the blankets on your body right now? These seem like such simple, automatic experiences that it's easy to assume nothing very interesting is going on, but the machinery underneath is extraordinarily complex and deeply strange. Let's start with vision, because it's the sense that neuroscientists have studied most extensively, and the story of how we understand it is one of the great intellectual adventures of the 20th century. Here's the basic setup. Light enters your eye through the pupil, gets focused by the lens, and hits the retina, which is a thin sheet of neural tissue lining the back of your eye. And the retina is remarkable. It's actually a piece of your brain that got pushed out during embryonic development and ended up inside your eyeball. It contains about 130 million photoreceptor cells, rods for dim light, cones for color and detail, and each one of those cells converts light into an electrical signal. So far, so straightforward. Light in, electrical signal out. But here's where it starts getting interesting. The signals from those 130 million photoreceptors don't just get passed straight to the brain as raw data. The retina itself does a tremendous amount of processing before the signal ever leaves the eye. There are several layers of neurons in the retina that combine, compare, and filter the raw photoreceptor signals. And by the time the processed information leaves the eye through the optic nerve, it's been compressed from 130 million photoreceptors down to about 1.2 million axons in the optic nerve. That's a compression ratio of more than 100 to 1. The retina is already deciding what's important and throwing away what isn't. The optic nerve carries those processed signals to the brain, where most of them end up in the primary visual cortex, which is located at the very back of your head. And in the 1950s and 1960s, two neuroscientists at Harvard, David Hubel and Torsten Wiesel, did a series of experiments on cats that revealed how the visual cortex processes information, and the results were so surprising and so beautiful that they fundamentally changed how we think about brain function. What Hubel and Wiesel did was insert a tiny electrode into the visual cortex of an anesthetized cat and record the activity of individual neurons while they showed the cat different visual stimuli on a screen. And what they found was that neurons in the visual cortex are astonishingly picky. They don't respond to just any visual stimulus. Each neuron responds best to a very specific feature of the visual scene. Some neurons fire like crazy when they detect an edge at a particular orientation, a vertical line, say. Tilt that line by 20 degrees and the neuron goes quiet. Other neurons respond specifically to horizontal lines, others to diagonal lines, others to edges moving in a particular direction at a particular speed. These are called simple cells, and they're the first stage of visual processing in the cortex. But it gets more complex as you move deeper into the cortex. You find neurons that respond to more abstract features, corners, for instance, or T-junctions, where two edges meet. And further along, you find neurons that respond to entire shapes, or to faces, or to specific objects. There's a region of the brain, in the temporal lobe, that responds specifically to faces. It's called the fusiform face area, and if it's damaged, you lose the ability to recognize faces, even though you can still see perfectly well. You can describe someone's features. They have blue eyes, dark hair, a long nose, but you can't put them together into a recognizable face. That condition is called prosopagnosia, or face blindness, and it tells you something profound about how the brain organizes information. Face recognition isn't just generic seeing. It's a dedicated system with its own neural hardware, built by evolution because recognizing faces was so important for our ancestors' survival. What Hubel and Wiesel had discovered was a hierarchy. The visual cortex builds complex representations from simple components, layer by layer. Edge-detecting neurons at the bottom feed into shape-detecting neurons in the middle, which feed into object-detecting neurons at the top. Simple features are assembled into complex percepts, step by step, each layer abstracting and combining the information from the layer below. And here's an amazing detail. This architecture is strikingly similar to the design of artificial neural networks used in modern AI for image recognition. The convolutional neural networks we talked about, the ones that power facial recognition in your phone and object detection in self-driving cars, were directly inspired by Hubel and Wiesel's discoveries about how the visual cortex processes information. The engineers literally copied the brain's architecture. Hubel and Wiesel won the Nobel Prize in 1981, and their work spawned an entire field of visual neuroscience. But here's the thing that always gets me about vision. The experience of seeing feels so simple, so immediate, so effortless. You open your eyes, and the world is just there. It doesn't feel like your brain is performing millions of computations to construct that visual scene from raw patterns of light hitting your retina. But it is. What you see is not reality. What you see is a model that your brain builds in real time from fragmentary and ambiguous data. Your brain fills in gaps, resolves ambiguities, makes assumptions, and constructs a coherent visual world that is, in a very real sense, a kind of controlled hallucination. That sounds dramatic, but it's accurate. The colors you see aren't properties of the objects out there. They're constructions of your visual system. The smoothness and continuity of your visual field is an illusion created by your brain stitching together information from eye movements you don't even notice. There's a blind spot in each eye where the optic nerve exits, and there are no photoreceptors, and your brain just fills it in. You never notice it. Your visual experience is a masterful piece of fiction that your brain writes and directs in real time, and it's so good that you mistake it for the script of reality itself. And hearing works in a similarly intricate way. Air pressure waves are captured by the eardrum, amplified by 3 tiny bones in the middle ear, the smallest bones in your body, and transmitted to the cochlea, a snail-shaped structure filled with fluid. Different frequencies cause different parts of the cochlea to vibrate. Hair cells detect these vibrations and convert them to electrical signals. The brain then separates the signal into component frequencies, identifies direction using tiny timing differences between your 2 ears, filters out background noise, and assembles all of this into the rich auditory world you experience. Every sense works on the same principle. A physical stimulus is converted by specialized receptor cells into electrical signals that travel to specific brain regions for processing. And that leads to one of the deepest questions in science, which we'll come to later. Why does any of this processing produce experience at all? That's the hard problem of consciousness, and it's still wide open. But let's stay with the machinery a while longer, because there's a crucial piece we haven't talked about: memory. How does the brain learn? How does it store information? And what, physically, changes in the brain when you remember something? This is one of the oldest questions in neuroscience, and for a long time, the answer was simply, we have no idea. People knew that memories existed, obviously, but what they were, physically, was a complete mystery. Was a memory a pattern of electrical activity, a chemical change, a structural alteration in the brain's wiring? In the early 20th century, a psychologist named Carl Lashley spent decades trying to find where memories were stored in the brain. He trained rats to run mazes, then surgically removed different parts of their cerebral cortex to see which removal would erase the memory, and he got a very puzzling result. No single brain region seemed to be the memory's home. He could remove large chunks of cortex, and the rat would still remember the maze, though it might be a bit slower or less accurate. The more cortex he removed, the worse the performance got, but it degraded gradually rather than disappearing all at once. Lashley concluded, somewhat despairingly, that memory was distributed throughout the cortex rather than localized in one spot. He famously wrote that after years of searching for the memory trace, he was sometimes tempted to conclude that learning was simply not possible. He was being ironic, obviously, but his frustration was real, and it pointed to something important. Memory isn't stored in a single location, the way a file is stored on a computer's hard drive. It's distributed across networks of neurons, embedded in the pattern of connections between them. And the physical basis of that storage, the actual mechanism by which connections change to encode new information, that wouldn't be understood for another several decades. The breakthrough came from a principle that had been proposed, in broad outline, remarkably early. In 1949, a Canadian psychologist named Donald Hebb published a book called The Organization of Behavior, and in it he proposed a deceptively simple idea about how learning might work at the level of individual synapses. Hebb's proposal, often paraphrased as neurons that fire together, wire together, went like this. When one neuron repeatedly participates in causing another neuron to fire, the connection between them gets stronger. The synapse becomes more efficient. the signal gets through more easily, and over time, this strengthened connection represents a memory, a learned association, a piece of knowledge encoded in the physical structure of the brain. Think about what that means. Every time you learn something, a new word, a new face, the layout of a new neighborhood, what's physically happening in your brain is that certain synaptic connections are getting stronger, while others are getting weaker. The pattern of strong and weak connections across billions of synapses is, in a very real sense, you. Your personality, your knowledge, your skills, your quirks, your fears, your favorite foods. All of it is encoded in the relative strengths of synaptic connections. You are, physically, a pattern of connections. Now, Hebb proposed this in 1949, but it took decades to find the actual cellular mechanism. And the person who found it, or at least who found the most important pieces of it, was a young Austrian-born neuroscientist named Erich Kandel. And the organism he used to study memory was not a rat or a monkey or a human. It was a sea slug. Specifically, a large, ungainly, not particularly beautiful marine snail called Aplysia californica. Kandel's choice of Aplysia is one of those decisions that looks eccentric at first glance, but turns out to be pure genius. He chose it for the same reason Hodgkin and Huxley chose the squid giant axon, because the neurons are enormous. Aplysia has only about 20,000 neurons, compared to our 86 billion, and some of those neurons are visible to the naked eye. You can identify individual neurons, come back to them day after day, and study their specific behavior. And Aplysia has simple behaviors that you can use to study learning. One of them is a defensive reflex. When you touch the siphon, a fleshy spout that the animal uses for breathing, it withdraws its gill. It's a protective reflex, like pulling your hand back from a hot stove. And this reflex can be modified by experience. If you repeatedly touch the siphon without doing anything harmful, the reflex weakens. The animal gets used to it, stops reacting. That's habituation, the simplest form of learning. On the other hand, if you pair the siphon touch with a strong electric shock to the tail, the reflex gets much stronger. The animal learns to react more vigorously. That's sensitization. And if you pair the touch and the shock in a specific way, you can produce classical conditioning. The animal learns to associate the touch with the shock and withdraws its gill even before the shock arrives. These are simple forms of learning, admittedly. Nobody is going to mistake Aplysia for Einstein. But the cellular mechanisms underlying them turn out to be remarkably similar to the mechanisms underlying more complex learning in more complex animals. What Kandel showed, over several decades of painstaking work, was that short-term memory and long-term memory are different at the molecular level. Short-term memory involves temporary changes in the strength of existing synaptic connections. Changes in the amount of neurotransmitter released, for instance. These changes last minutes to hours and don't require the cell to make new proteins. They're modifications of existing machinery. Long-term memory, on the other hand, requires something more permanent. To convert a short-term memory into a long-term one, the neuron has to activate genes in its nucleus, manufacture new proteins, and use those proteins to physically remodel the synapse, growing new synaptic connections, enlarging existing ones, making structural changes that are durable. This is why memories consolidate over time, why sleep is important for memory formation. During sleep, the brain appears to replay and strengthen the synaptic patterns that were activated during the day, and why a blow to the head can wipe out recent memories while leaving old ones intact. Recent memories haven't been fully consolidated yet. They're still in the fragile, short-term phase. Old memories have been structurally embedded in the brain's wiring. Kandel won the Nobel Prize in Physiology or Medicine in 2000 for this work, and the principle he helped establish, that learning and memory are fundamentally about changes in synaptic strength, has become one of the central tenets of modern neuroscience. It's called synaptic plasticity, and it's the brain's most remarkable property. The brain is not a fixed circuit like a computer chip. It's a living, constantly rewiring system that reshapes itself in response to experience. Every conversation you have, every book you read, every skill you practice is literally, physically, changing the structure of your brain. The brain you have right now, tonight, is different from the brain you had this morning because the day's experiences have altered some fraction of your 100 trillion synaptic connections. And this brings us to a phenomenon that was discovered in 1973 and has become one of the most intensely studied topics in all of neuroscience. It's called long-term potentiation, or LTP, and it's considered the primary cellular mechanism of learning and memory in the mammalian brain. Two researchers, Timothy Bliss and Terje Lomo, working with rabbit hippocampi in a laboratory in Norway, found that if you stimulate a neural pathway with a rapid burst of high-frequency electrical pulses, the synapses in that pathway become stronger, and they stay stronger for hours, days, or even weeks. A brief burst of intense activity produces a lasting change in synaptic strength. That's exactly what Hebb had predicted back in 1949. Neurons that fire together wire together, and Bliss and Lomo had found the physiological basis for it. The hippocampus, by the way, is where they found LTP, and that's not a coincidence. The hippocampus is a seahorse-shaped structure deep inside the temporal lobe, and it plays a crucial role in forming new memories. We know this because of one of the most famous patients in neuroscience history, a man known for decades only by his initials, HM. His real name was Henry Molaison, and his story is both illuminating and heartbreaking. In 1953, Henry Molaison was a young man suffering from severe, debilitating epilepsy. His seizures were so frequent and so violent that he couldn't hold a job or live a normal life. The seizures were traced to his temporal lobes, and in desperation, a neurosurgeon named William Scoville removed the hippocampus and surrounding structures from both sides of Henry's brain. The surgery worked. The seizures were largely controlled, but the consequences were devastating. Henry lost the ability to form new long-term memories. He could remember his childhood. He could remember events from before the surgery. His intelligence was intact. His personality was unchanged. He could carry on a perfectly normal conversation. But the moment a conversation ended and you left the room, he forgot you existed. He could meet the same person every day for 30 years and never recognize them. He could read the same magazine over and over, each time finding it completely new. He was stuck in a perpetual present, unable to lay down new episodic memories. Think about what that tells you. The hippocampus isn't where memories are permanently stored. Henry's old memories were intact. But the hippocampus is essential for creating new ones, for converting experiences into lasting memories. It's like a gateway. Information has to pass through the hippocampus to be stamped into the cortex as a long-term memory, and without that gateway, the stamp never gets applied. The experience happens, it's processed, it's conscious, but it doesn't stick, it evaporates. Henry Molaison was studied for over 50 years by the neuroscientist Brenda Milner and her students, and the data from his case reshaped our understanding of memory. He died in 2008 at the age of 82, and his brain was carefully preserved and sliced into over 2,000 sections for detailed anatomical study. He contributed more to our understanding of memory than perhaps any other single individual, and he spent most of his life unable to remember any of it. Now, here's something subtle and important about the hippocampus and memory that Henry's case helped reveal. Henry couldn't form new episodic memories of specific events and experiences, but he could still learn certain kinds of things. Specifically, he could learn motor skills. If you taught him to trace a pattern while looking at his hand in a mirror, a task that's surprisingly tricky, he would get better at it over multiple days, even though he had no memory of ever practicing. Every session, he'd say he'd never done it before, but his performance improved steadily. This showed that there are different memory systems in the brain, handled by different neural structures. Episodic memory, remembering what happened, depends on the hippocampus. Procedural memory, learning how to do things, depends on other structures, including the basal ganglia and the cerebellum. You can lose one without losing the other. This is why people with Alzheimer's disease, which devastates the hippocampus early on, can often still play the piano or ride a bike even when they can't remember where they are or who you are. The motor skill is stored in a different system from the biographical memory. So here's the picture we've built up. The brain is made of billions of individual neurons. Those neurons communicate through a combination of electrical signals within the cell and chemical signals at the synapse. The strength of synaptic connections can change with experience, which is how the brain learns and remembers. Different brain regions handle different functions. The visual cortex processes vision. The hippocampus is essential for forming new memories. The motor cortex controls movement. The auditory cortex processes sound. And all of this is running simultaneously, in parallel, producing the seamless experience of being alive and conscious in the world. But here's a question we haven't addressed yet, and it's a big one. Where did all of this come from? How did brains evolve? Because the brain you carry around, with its 86 billion neurons and its cortical hierarchies, and its memory systems, and its capacity for language and abstract thought, didn't just appear out of nowhere. It was built layer by layer over hundreds of millions of years of evolution, and the story of how simple nervous systems became complex brains is one of the most fascinating narratives in all of biology. And it starts not with neurons at all, but with something much simpler, just a single cell trying to get by in an ancient ocean. Think about the very earliest forms of life on Earth. Single-celled organisms floating in ancient seas, roughly 3.5 billion years ago. No brain, no nervous system, not even a nervous cell. And yet, even these simplest creatures had to solve a fundamental problem: how to respond to the environment, how to move toward food and away from danger, how to detect changes in light, temperature, and the chemical composition of the surrounding water. Bacteria can do this. An E. coli bacterium, which doesn't have a single neuron, can detect the concentration of sugar in its environment and swim toward higher concentrations. It does this through a simple mechanism called chemotaxis. Receptors on its surface detect the chemical, and a signaling cascade inside the cell controls the rotation of its flagella, steering it up the concentration gradient. It's not thinking. It's not deciding. It's a chemical reflex, a molecular machine responding automatically to its environment. But it's solving the same basic problem that brains solve, just with much simpler machinery. Sense the world, respond appropriately, survive. And this is the key insight about brains. They didn't arise from nothing. They evolved from simpler sensory and response systems, gradually becoming more complex, more centralized, more capable, over millions of generations. The brain is not some magic organ that appeared suddenly in the history of life. It's the latest iteration of a very old solution to a very old problem: how to navigate a changing world. Now, when did actual nerve cells first appear? We don't know exactly, but we can look at the simplest living animals that have them, and the best candidates are cnidarians, jellyfish, sea anemones, corals, and their relatives. These animals diverged from our lineage something like 600 million years ago, and they have nervous systems, but nothing you'd call a brain. A jellyfish has what's called a nerve net, a distributed network of neurons spread throughout its body, with no central processing point. There's no boss, no command center. Each neuron connects to its neighbors, and signals spread outward from the point of stimulation in all directions, like ripples from a stone thrown in water. This is enough for what a jellyfish needs to do: contract its bell to swim, extend its tentacles to capture prey, respond to touch and light and gravity. It's a decentralized system, and it works for a simple, radially symmetric body plan. But here's the thing: to do anything more complex, to have directed movement, to integrate different senses, to make something resembling a decision, you need centralization. You need a place where information comes together and is processed in a coordinated way. And this is where the story of the brain really begins. The first animals to develop anything like a centralized nervous system were probably bilateral animals, creatures with a front end and a back end, a top and a bottom, a left side and a right side. This body plan evolved somewhere around 550 to 600 million years ago, during or just before the Cambrian explosion, that remarkable period when nearly all the major animal body plans appeared in the fossil record over a geologically brief span of time. And bilateral symmetry created a new problem and a new opportunity. If you have a front end, that's the part that encounters the environment first. That's the part that needs to sense what's coming. So sensory organs tend to cluster at the front: eyes, antennae, chemical receptors. And if the sensory organs are at the front, it makes sense to put the neural processing close to them, to minimize the distance signals have to travel. So neurons began to cluster at the front end too, forming a concentration of neural tissue that would eventually become a brain. This process is called cephalization, the development of a head. The simplest example of this you can see today is in flatworms, those thin, elegant little animals that glide along the bottoms of ponds and streams. A flatworm, like a planarian, has two clusters of neurons at its front end, two tiny ganglia, essentially miniature brains, connected to a pair of nerve cords that run down the length of its body. It has two primitive eye spots that can detect light, and it can learn. If you train a planarian to associate light with an electric shock, it will begin to avoid the light. That's associative learning, the same basic phenomenon that Pavlov demonstrated with dogs, happening in an animal with maybe 20,000 neurons and no structure that anyone would dignify with the word brain. But the seeds are there. Sensory organs at the front, clustered neurons nearby, nerve cords carrying signals to the body. This is the body plan that evolution would elaborate and refine for the next 500 million years. The insects took this plan and ran with it. A honeybee's brain contains about 1 million neurons, but with those 1 million neurons, it can fly with exquisite precision, navigate kilometers from the hive, communicate food locations through the waggle dance, and learn associations between odors and rewards, all with a brain smaller than the head of a pin. Evolution has had hundreds of millions of years to optimize insect nervous systems, and the result is astonishing computational power packed into minimal space. But our lineage runs through the vertebrates, and the vertebrate brain has its own distinctive architecture tracing back to our fish ancestors. The earliest vertebrates were jawless fish, something like modern lampreys, swimming in the oceans around 500 million years ago, and they already had the basic vertebrate brain layout that persists, modified and elaborated, in every vertebrate alive today, including you. The brain was divided into three main sections. The hindbrain, at the back, controlled basic functions like breathing, heart rate, and balance. The midbrain, in the middle, processed sensory information, especially vision. And the forebrain, at the front, handled smell and some basic behavioral regulation. Here's the thing about evolution. It rarely starts from scratch. It builds on what's already there. So as vertebrates evolved over hundreds of millions of years, the brain didn't get redesigned. It got expanded. New layers of tissue grew on top of old ones. New structures were added, but the old ones remained, still doing their jobs. This is why the neuroscientist Paul MacLean proposed his famous triune brain model in the 1960s, the idea that the human brain is essentially three brains layered on top of each other. The reptilian brain at the core, handling basic survival functions. The limbic system wrapped around it, handling emotions and memory. And the neocortex on the outside, handling higher cognition, language, and abstract thought. Now, this model is a dramatic oversimplification, and modern neuroscientists have pointed out many ways in which it's misleading. The boundaries between these systems are not as clean as MacLean suggested, and non-mammalian vertebrates have more sophisticated cognitive abilities than the model implies. But the basic insight that the human brain retains ancient structures overlaid with newer ones is essentially correct. When you feel a surge of fear or anger, when your heart races in response to danger, that's circuitry that your ancestors shared with lizards. When you feel the warm glow of cuddling with a loved one, that's circuitry that evolved in early mammals. When you compose a sentence or contemplate the future, that's the neocortex, the latest addition. The great story of mammalian brain evolution is the expansion of the cerebral cortex. In reptiles, the cortex is thin, a few layers of cells. In mammals, it expanded dramatically, both in thickness. The mammalian neocortex has 6 distinct layers of cells, compared to 3 in reptiles, and in total surface area. And this expansion happened most dramatically in the primate lineage. Our primate ancestors, living in trees, needed sharp vision, precise hand-eye coordination, and the ability to navigate complex social groups. These pressures drove the expansion of the visual cortex, the motor cortex, and especially the frontal cortex, which is involved in planning, decision-making, and social cognition. And then, in the hominin lineage, the lineage that includes humans and our recent ancestors, something extraordinary happened. The brain underwent one of the most dramatic expansions in the history of life on Earth. Over the course of about 2 to 3 million years, the hominin brain roughly tripled in size. Australopithecus, our distant ancestor from about 3 million years ago, had a brain of around 400 cubic centimeters, roughly the size of a chimpanzee's. Homo erectus, about 1.5 million years ago, had a brain of around 900 cubic centimeters. And modern Homo sapiens, appearing around 300,000 years ago, have brains averaging about 1,350 cubic centimeters. That tripling happened remarkably fast, in evolutionary terms, and it came with enormous costs. Think about the costs. The brain accounts for about 2% of your body weight, but consumes about 20% of your calories. In a newborn, it's even more. About 60% of the infant's energy goes to running the brain. A bigger brain also means a bigger skull, which makes human birth more dangerous than in almost any other animal. Babies are born with their brains not yet fully developed, which is why human infants are so helpless compared to other species. A newborn horse can stand within hours. A newborn human can't hold its own head up. So the human brain is expensive, difficult to birth, and takes years to reach maturity. Evolution doesn't produce extravagances without reason. The payoff was intelligence, the ability to plan, cooperate, communicate, make tools, and pass knowledge through language. These capabilities gave our ancestors such a massive survival advantage that the costs were more than worth it. But here's an important nuance. Brain size alone doesn't determine intelligence. If it did, elephants and whales, which have brains much larger than ours, would be the smartest animals on the planet. What matters more is the ratio of brain size to body size, the encephalization quotient, as it's called. And in this measure, humans are off the charts. Our brains are about 7 times larger than you'd predict for a mammal of our body size. Dolphins are about 4 times. Chimpanzees are about twice, and some of the most cognitively impressive birds, like crows and parrots, have brains that are much smaller than ours in absolute terms, but remarkably large relative to their body size, and packed with neurons at higher densities than mammalian brains. A raven, with a brain weighing about 15 grams, can use tools, plan for the future, recognize itself in a mirror, and solve multi-step problems that stump many mammals with much larger brains. This brings up something really interesting: the idea that intelligence can evolve independently through different neural architectures in very different lineages. Birds evolved from dinosaurs. Their last common ancestor with mammals lived over 300 million years ago, and yet crows and parrots have convergently evolved cognitive abilities that rival those of great apes. They've done it with a completely different brain structure. Birds don't have a layered neocortex the way mammals do. Their cognition is powered by a structure called the pallium, which is organized differently, but can apparently perform many of the same functions. It's as if evolution found 2 different engineering solutions to the same design challenge, 2 ways to build a thinking machine. And this convergent evolution of intelligence is something we see across the animal kingdom, in places you might not expect. Octopuses, for example. An octopus is a mollusk. Its closest relatives are clams and snails, and yet octopuses are remarkably intelligent. They can solve puzzles, open jars, use coconut shells as portable shelters, learn by observing other octopuses, and apparently play. There are reports of octopuses squirting jets of water at floating bottles just to watch them bob around, which looks an awful lot like play behavior. An octopus has about 500 million neurons, which is more than most vertebrates except mammals and birds. But here's what's really wild. Most of those neurons are not in the octopus's brain. They're in its arms. Each arm has its own mini-brain, its own cluster of ganglia that can control the arm's movements semi-independently. An octopus can be solving a problem with one arm, while another arm is exploring a crevice, and a third is opening a jar. The cognitive architecture is radically different from anything in the vertebrate world, and yet the behavioral output is impressively sophisticated. So intelligence isn't a single solution that evolution found once. It's a problem that's been solved repeatedly, in different ways, in different lineages, using different neural architectures. And that tells us something deep about the nature of brains. The specific wiring diagram matters less than the computational principles. Different hardware can run similar software. What those computational principles actually are, and whether they can be captured in artificial systems, is one of the great questions that modern neuroscience and artificial intelligence are both trying to answer from different directions. But let's come back to the human brain for a moment, because there's a feature of it that we haven't talked about yet, and it may be the most important one of all: language. The capacity for complex, recursive, open-ended language. And the story of how we figured out where language lives in the brain starts with a French physician, a patient who could only say one word, and a debate about localization that played out in the heated lecture halls of 19th century Paris. The physician's name was Paul Broca, and the patient's name was Louis Victor Leborgne, though history remembers him by the only word he could say: tan. Leborgne had been a patient at the Bicêtre Hospital in Paris for over 20 years. He had lost the ability to speak when he was around 30 years old. He could understand language perfectly well. He could follow conversations, respond to questions with gestures, and his intelligence seemed unimpaired. But when he tried to speak, the only syllable that came out was tan, over and over. Tan, tan, tan. Sometimes he'd add a swear word. There's a charming detail that tells you he was still in there, still feeling things, still frustrated. But meaningful speech was gone. In 1861, Leborgne came to Broca's surgical ward for an unrelated condition, and shortly after, he died. Broca performed an autopsy and examined his brain. And there, on the left side of the frontal lobe, was a region of clear damage, an area where the tissue had been destroyed, probably by a stroke years earlier. Broca examined other patients with similar speech difficulties and found similar damage, always in the same area, always on the left side. And he concluded that this specific region of the left frontal lobe was responsible for the production of speech. We now call it Broca's area, and damage to it produces what's called Broca's aphasia, an inability to produce fluent speech, even though language comprehension remains intact. A few years later, a German neurologist named Karl Wernicke identified a different kind of language problem in patients with damage to a different brain region. These patients could speak fluently, their words flowed freely, with normal rhythm and intonation. But what they said didn't make sense. They would produce grammatically correct sentences filled with nonsense words, or with real words arranged in meaningless combinations, and they couldn't understand what was said to them. The damage was in the left temporal lobe, in an area now called Wernicke's area. Wernicke's aphasia is almost the mirror image of Broca's. The speech flows, but the meaning is gone, and comprehension is severely impaired. Together, these discoveries established something remarkable. Language isn't a single ability handled by a single brain region. It's a complex skill that depends on multiple specialized areas working together. Broca's area handles the production of speech, the motor planning of how to move the mouth, tongue, and larynx to form words. Wernicke's area handles comprehension, the understanding of language's meaning. And a thick bundle of nerve fibers called the arcuate fasciculus connects the two areas, allowing production and comprehension to coordinate. But here's the really strange part. In the vast majority of people, both Broca's area and Wernicke's area are on the left side of the brain. Language is lateralized. It lives predominantly in one hemisphere, and this lateralization is one example of a broader phenomenon in brain organization. The two hemispheres of the cerebral cortex look nearly identical from the outside, but they are not functionally interchangeable. The left hemisphere, in most people, is dominant for language, logic, and analytical processing. The right hemisphere tends to be more involved in spatial reasoning, face recognition, emotional processing, and holistic thinking. The popular culture version of this, left brain logical, right brain creative, is a massive oversimplification. But the underlying asymmetry is real, and some of the most dramatic evidence comes from patients who have had the connection between their hemispheres surgically severed. The corpus callosum, a massive bundle of about 200 million nerve fibers, connects the two hemispheres. A handful of epilepsy patients had it cut to prevent seizures from spreading, and these split-brain patients were studied extensively by Roger Sperry and Michael Gazzaniga, with astonishing results. Here's the setup. Because of how the visual system is wired, information from your left visual field goes to the right hemisphere, and information from your right visual field goes to the left hemisphere. Normally, the corpus callosum immediately shares this information between the hemispheres, so you don't notice the split. But in a split-brain patient, that sharing can't happen. Each hemisphere only knows about what's in its visual field. So imagine you flash a word, let's say hammer, in the left visual field of a split-brain patient. That information goes to the right hemisphere, which, in most people, doesn't have language. If you ask the patient what they saw, they'll say nothing. Their left hemisphere, which does have language and is the one doing the talking, genuinely didn't see it. But if you ask the patient to reach behind a screen with their left hand, which is controlled by the right hemisphere, and pick out the object that matches what they saw, they'll correctly pick up a hammer. The right hemisphere saw it, recognized it, and can act on it. It just can't tell you about it in words, because words are the left hemisphere's job. It's as if there are two separate conscious minds in one skull, each aware of different things, each controlling different parts of the body. Sperry won the Nobel Prize in 1981 for this work, and it raised profound questions about the nature of consciousness, identity, and selfhood that are still debated today. If you split the brain, do you split the person? Are there now two conscious entities in one body? The patients themselves mostly reported feeling normal, because in everyday life, both eyes are open and both hemispheres get similar information. The strange dissociations only appeared under carefully controlled laboratory conditions, but the implications are unsettling. Our sense of being a single, unified self may depend on a physical connection, a bundle of fibers, that can be cut with a surgeon's blade. For most of neuroscience's history, studying the brain meant studying it after something had gone wrong, a stroke, an injury, surgery. You'd find a patient with damage to a specific region and infer what that region did. But there's an obvious limitation. You're always working backward from damage, and brain damage is never clean or precise. What neuroscience desperately needed was a way to watch the brain in action, in a healthy, living brain. And in the last few decades, that's exactly what we've gotten. The revolution in brain imaging technology has transformed neuroscience more profoundly than any development since the Golgi stain. The real game-changer came in 1990, when Seiji Ogawa at Bell Laboratories discovered that MRI machines could detect changes in blood oxygenation in the brain. When neurons in a region become active, they consume more oxygen, and the blood changes its magnetic properties in ways MRI can detect. This is functional MRI, or fMRI, and it allows you to create maps of brain activity with spatial resolution of a few millimeters. It's not perfect. It's measuring blood flow, an indirect proxy for neural activity. But it's been extraordinarily revealing. With fMRI, researchers have been able to map the brain's functional organization with unprecedented detail. They've identified specialized regions for recognizing faces, processing places, understanding numbers, empathizing with others, even regions that respond specifically to pictures of food. They've shown that the brain is organized into large-scale networks that cooperate across distant regions, connected by long-range bundles of nerve fibers. One of the most fascinating discoveries is the default mode network, a set of brain regions that become active not when you're performing a task, but when you're doing nothing, resting, daydreaming, mind-wandering. This network, which includes parts of the medial prefrontal cortex, the posterior cingulate cortex, and the lateral temporal cortex, appears to be involved in self-referential thinking, imagining the future, remembering the past, and thinking about other people's mental states. It's the brain's idle mode, and it's remarkably active. Your brain, when it's ostensibly doing nothing, is doing a tremendous amount of internal processing. It's running simulations, rehearsing scenarios, consolidating memories, constructing narratives. The resting brain is not resting at all. This is the beginning of what's now called connectomics, the ambitious project to map the complete wiring diagram of the brain. The most complete connectome so far is from a tiny worm called C. elegans, which has exactly 302 neurons and about 7,000 synaptic connections, mapped completely by Sidney Brenner's team in 1986. Having the complete wiring diagram of an entire nervous system has been invaluable. But the human brain? 86 billion neurons, 100 trillion synapses. In 2024, a Harvard and Google team mapped just 1 cubic millimeter of human cortex, about the size of a grain of sand, and found 57,000 cells and 150 million synapses. The dataset was over a petabyte, a grain of sand's worth of brain, and it takes 1 million gigabytes to describe. And we're not just mapping, we're manipulating. One of the most powerful tools in modern neuroscience is optogenetics, a technique developed in the early 2000s, primarily by Carl Diceroth at Stanford and Ed Boyden at MIT. Optogenetics allows researchers to control specific neurons with light. Here's how it works. You genetically modify a specific type of neuron so that it produces a light-sensitive protein, usually borrowed from algae, which use similar proteins to detect light. Then you implant a tiny fiber optic cable into the brain, right next to the neurons you've modified. When you shine light through the fiber, the modified neurons fire. Turn off the light, and they stop. For the first time, you can activate or silence specific populations of neurons with millisecond precision and watch what happens to the animal's behavior. The results have been remarkable. Using optogenetics in mice, researchers have turned fear responses on and off like a switch. They've implanted false memories. They've made mice run in circles, eat when they're not hungry, fall asleep, and fight with other mice, all by stimulating specific neural circuits with light. It's an extraordinarily precise tool, and it's given neuroscience something it never had before: the ability to test causal relationships between brain activity and behavior. With fMRI, you can see that a brain region is active during a task, but you can't be sure it's causing the behavior. It might just be along for the ride. With optogenetics, you can directly activate that region and see if the behavior follows. If it does, you've established a causal link. Diceroth has also developed a technique called clarity, which makes brain tissue transparent while preserving its structure. You can hold a mouse brain in your hand and see right through it, all the neurons and their connections visible in 3 dimensions. It sounds like science fiction. It's real. And it's part of a larger trend in neuroscience, the development of tools that would have seemed impossible a generation ago. Tools that are allowing us to study the brain at a level of detail that Cahal could only dream of. And yet, for all these remarkable tools, for all the maps and measurements and manipulations, there's a sense in the field, a widely shared sense, I think, that we're still missing something fundamental. We have more data about the brain than at any point in history. We can image it, map it, manipulate it, simulate parts of it. But we still don't really understand how it works, not in the deep sense. We don't understand how the coordinated activity of billions of neurons gives rise to a thought. We don't understand how the firing of cells produces the experience of seeing red, or feeling sad, or understanding a joke. We have pieces of the puzzle, many of them beautiful and precise, but the big picture remains elusive. And this gap between data and understanding brings us to what is probably the deepest and most mysterious question in all of science: consciousness. What is it? Where does it come from? And why does it exist at all? Here's the puzzle, stripped down to its simplest form. You are a collection of atoms. Those atoms are arranged into molecules. Those molecules form cells. Among those cells are neurons, which process information through electrical and chemical signals, as we've been discussing all night. And somehow, out of all that physical machinery, all those ions flowing through channels, all those neurotransmitters crossing synapses, all those action potentials propagating down axons, somehow there arises something utterly unlike anything else in the known physical world. There arises experience. The felt quality of being you, the redness of red, the taste of coffee, the ache of loneliness, the sensation, right now, of lying in the dark, breathing, thinking, something it's like to be you. This is what philosophers call the hard problem of consciousness, a term coined by the philosopher David Chalmers in 1995. And it's called the hard problem to distinguish it from what Chalmers called the easy problems, which are, to be fair, not easy at all. The easy problems are things like explaining how the brain processes information, how it controls behavior, how it discriminates sensory stimuli, how it integrates information from different sources. These are the problems we've been talking about all night, and they're formidable scientific challenges. But at least we know what kind of answers to expect. They're problems of mechanism. You study the neurons, the circuits, the chemicals, and you figure out how the machine works. The hard problem is different. The hard problem is this: why does any of that processing feel like anything? Why isn't the brain just a biological computer, processing information in the dark, producing behavior without any accompanying experience? You could imagine a being that behaves exactly like you in every way, responds to stimuli, holds conversations, reports being conscious, but has no inner experience at all, no felt quality, no light on inside. Philosophers call this a zombie, and the hard problem is essentially the question of why we aren't zombies. Why is there an inner life? And the honest answer, right now, is that nobody knows. This isn't one of those questions where scientists are closing in and the answer is just around the corner. We genuinely don't have a theory of consciousness that most researchers agree on. We don't even agree on what form such a theory should take. Some neuroscientists think consciousness will eventually be explained by understanding the brain's information processing in sufficient detail, that it's an emergent property of complex neural computation, the way wetness emerges from the collective behavior of water molecules, even though no single molecule is wet. Others think we need fundamentally new concepts, new physics even, to explain it. And some philosophers have argued that consciousness may be a basic feature of the universe itself, not something that emerges from physical processes, but something that's always been there, woven into the fabric of reality at a fundamental level. This view is called panpsychism, and while it sounds exotic, it's been taken seriously by respected thinkers throughout history and is experiencing a genuine resurgence in philosophical circles today. But let me be careful here, because I don't want to give the impression that neuroscience has nothing to say about consciousness. It has plenty. We just haven't solved the hard problem. What neuroscience has done is identify many of the neural correlates of consciousness, the brain states that are associated with conscious experience. We know, for instance, that consciousness seems to require activity in the cerebral cortex and the thalamus, a relay structure deep in the brain that sends information to the cortex and receives it back. Damage to certain parts of the thalamus can produce a permanent loss of consciousness, even though the brainstem keeps the body alive. We know that general anesthetics, which abolish consciousness temporarily, appear to work partly by disrupting the communication between different brain regions rather than simply shutting them all down. And we know that certain patterns of brain activity are associated with awareness and others aren't. When information is processed unconsciously, it activates certain brain regions but not others, and it doesn't trigger the same kind of widespread, coordinated activity across the cortex that conscious processing does. There are serious scientific theories trying to crack this. Giulio Tononi's integrated information theory proposes that consciousness corresponds to a mathematical quantity called phi, which measures how much integrated information a system generates. The more a system is both differentiated and integrated, many possible states, all bound together, the more conscious it is. Another influential framework, global workspace theory, proposed by Bernard Bars and elaborated by Stanislas Dehaene, likens consciousness to a global broadcast system. Most brain processing happens unconsciously, but when information becomes important enough, it gets broadcast to widely distributed brain regions, making it available for report, memory, and action. That broadcast is consciousness. Dehaene has shown that unconsciously processed stimuli activate only local brain regions, while consciously perceived stimuli trigger a sudden, widespread ignition of activity across the cortex. There's an all-or-nothing transition between unconscious processing and conscious awareness. Now, all of this brings up a question that is both scientifically fascinating and ethically crucial. If consciousness is a product of brain activity, and if different brains produce different levels and types of consciousness, then what about other animals? Are animals conscious? Do they have inner experiences? Does a dog feel joy when it sees its owner? Does a fish feel pain when it's hooked? Does a bee have some dim flicker of experience as it navigates from flower to flower? The scientific answer, as honestly as I can state it, is that we believe most vertebrates are almost certainly conscious to some degree, but we don't know exactly what their experience is like. The neural machinery for consciousness, the cortical and thalamic circuits, the neurotransmitter systems involved in arousal and attention, the neural correlates of consciousness that we've identified in humans. These are largely shared across mammals and, in modified form, across all vertebrates. In 2012, a group of prominent neuroscientists signed the Cambridge Declaration on Consciousness, which stated that the weight of evidence indicates that humans are not unique in possessing the neurological substrates that generate consciousness, and that non-human animals, including all mammals and birds, and many other creatures, including octopuses, also possess these neurological substrates. This doesn't mean a dog experiences the world the same way you do. A dog's consciousness is presumably shaped by its sensory abilities. Its extraordinary sense of smell creates a dimension of experience that you and I can barely imagine. A bat that navigates by echolocation experiences a sonar world that has no analog in human experience. The philosopher Thomas Nagel wrote a famous paper in 1974 called "What Is It Like to Be a Bat?" and his point was precisely this. There is something it is like to be a bat, some subjective experience, but we may never be able to fully understand what that experience is, because it's so alien to our own sensory world. But even if we can't know exactly what an animal experiences, the evidence that many animals do experience something, that they feel pain, fear, pleasure, perhaps even something like grief, has profound ethical implications. And the neuroscience is increasingly clear on this. The brain structures and neurotransmitter systems involved in emotional processing are evolutionarily ancient. They're shared across mammals and, in varying forms, across vertebrates more broadly. The neuroscientist who established much of this was Jaak Panksepp, who identified 7 basic emotional circuits in the mammalian brain, each grounded in specific brain structures: seeking, rage, fear, lust, care, panic, grief, and play. And here's a charming detail. Panksepp discovered that rats laugh. When you tickle a young rat, it produces ultrasonic vocalizations at around 50 kilohertz, far above human hearing. Rats that are tickled seek out more tickling. They make these calls during play with other rats. Panksepp argued, with considerable evidence, that these are the functional equivalent of laughter, an expression of joy associated with social play. Does a rat experience joy the way you do? Probably not. But the basic emotional machinery, the circuits that generate pleasure and distress, these are ancient and shared across mammals. When an animal is in pain, the same brain regions that process pain in humans light up. The neurotransmitters are the same. The evolutionary continuity is clear, and this continuity extends beyond mammals. Fish have nociceptors, sensory receptors that detect tissue damage, and they respond to painful stimuli with behavioral and physiological changes that look a lot like pain responses in mammals. They'll avoid places where they've been hurt, and they'll reduce their food intake. They'll show elevated cortisol. Whether this constitutes felt pain, in the sense of conscious suffering, is genuinely debated. But the trend in the science has been steadily in the direction of attributing more, not less, inner life to non-human animals. And this brings us back, beautifully, to the question of how brains work, because the reason we care about how brains work is not just intellectual curiosity, as delightful as that is. We care because brains are what produce experience. Brains are what make organisms care about things. A rock doesn't mind being kicked. An earthworm might? A dog almost certainly does. And the difference lies entirely in what the brain is doing, in the patterns of activity reverberating through neural circuits, in the neurotransmitters flooding across synapses, in the integrated processing that, somehow, gives rise to the felt quality of being alive. And yet, somehow, out of this unfathomable complexity, 86 billion neurons, 100 trillion synapses, more possible brain states than there are atoms in the observable universe, emerge the orderly patterns that constitute thought, perception, emotion, memory, language, consciousness. This 3-pound organ, consuming 20 watts of power, less than a dim light bulb, does things that no computer can yet match. It recognizes faces in a fraction of a second. It understands language with its infinite nuances. It generates creative ideas. It feels. It dreams. It falls in love. How? We don't fully know. And that's what makes neuroscience so exciting right now. The tools exist to study the brain at levels of detail that would have seemed like science fiction a generation ago. Optogenetics, fMRI, connectomics, 2-photon imaging. The rate of discovery is accelerating, and yet the deepest questions remain open. But that's the future. Right now, tonight, let's sit with what we've learned, because the story is, in its own right, extraordinary. We started with the ancient Egyptians pulling brains out through the nose and throwing them away. And look where we've gotten. We now know that the brain is made of billions of individual neurons, each one a tiny electrochemical device that converts inputs into outputs through the flow of ions across its membrane. We know that these neurons communicate at synapses, using chemical messengers that can excite or inhibit the next cell. We know that the strength of these synaptic connections changes with experience, which is how the brain learns and remembers. We know that different brain regions are specialized for different functions: vision in the back, motor control on the top, language on the left side, emotion and memory deep inside. We know that the brain evolved from the simplest nerve nets in jellyfish to the extraordinary complexity of the human cerebral cortex over hundreds of millions of years, adding new layers of circuitry on top of old ones, each addition providing new capabilities while preserving the ancient systems underneath. And we know the stories of the people who figured all of this out. Galvani, making dead frogs' legs twitch with electricity. Golgi, staining neurons in a hospital kitchen by candlelight. Cajal, spending decades at his microscope, drawing neurons with an artist's hand and a scientist's eye, proving that the brain is made of individual cells. Hodgkin and Huxley, threading electrodes into squid giant axons and deciphering the mechanism of nerve impulses. Otto Loewy, waking from a dream at 3 a.m. and rushing to his laboratory to prove that synaptic transmission is chemical. Eric Kandel, studying memory in sea slugs and finding that learning physically rewires the brain. Hubel and Wiesel, discovering that individual neurons in the visual cortex are tuned to specific features of the visual world. Henry Molaison, who lost the ability to form new memories and, in doing so, taught us more about memory than perhaps any other person who ever lived. These are human stories, stories of curiosity and persistence, and sometimes of terrible sacrifice. And they've given us something genuinely remarkable, a growing understanding of the organ that makes us who we are. But I want to talk about a few more things before we let the night take over, because the brain does some things that are especially relevant to you right now, lying here in the dark. And the first one is sleep. Sleep is, when you think about it, one of the most bizarre things animals do. You lose consciousness. You become unresponsive to your environment. You stop doing anything productive. You're vulnerable to predators. And you do this for about a third of your life. Every animal that has a brain sleeps, or at least enters something resembling sleep, even fruit flies, even roundworms. Sleep is so universal, so deeply conserved across evolution, that it must serve some essential function. An animal that evolved to skip sleep would have a massive competitive advantage: more time to eat, mate, and watch for predators. But no animal has ever evolved to skip sleep. Whatever sleep does, it's apparently so important that giving it up isn't an option. And for most of the history of neuroscience, nobody could explain why. Sleep was, as the sleep researcher Alan Rechtschaffen once put it, the biggest open question in biology. We knew that sleep deprivation was devastating. Rats kept awake continuously die within a few weeks, and humans who are sleep-deprived show severe cognitive impairment, emotional instability, and eventually something like psychosis. But what sleep was actually doing that made it so essential was a mystery. In recent years, though, that mystery has started to crack open, and the answers are fascinating. One major function of sleep appears to be memory consolidation. Remember how we talked about the hippocampus and its role in forming new memories? During sleep, the hippocampus appears to replay the day's experiences, sending them to the cortex in a compressed, accelerated form. Neurons that were active during a waking experience fire again during sleep, in the same patterns, as if the brain is rehearsing and reinforcing what it learned. This is particularly true during a stage of sleep called slow-wave sleep, when large, slow electrical waves sweep across the cortex. These waves seem to provide a window during which synaptic connections can be modified and memories can be transferred from the hippocampus to the cortex for long-term storage. Studies have shown that sleeping after learning significantly improves memory retention compared to staying awake for the same amount of time. The brain isn't just resting during sleep, it's studying. And then there's REM sleep, rapid eye movement sleep, the stage when most dreaming occurs. During REM sleep, the brain is remarkably active. The cortex shows patterns of activity that look similar to wakefulness, but the body is essentially paralyzed, a clever arrangement that prevents you from acting out your dreams. The exact function of REM sleep is still debated, but it seems to be important for emotional processing and for the kind of creative, associative thinking that connects distant ideas. There's evidence that REM sleep helps with problem solving, with emotional regulation, and with the brain's ability to form flexible connections between concepts. It's the brain's wild card, its experimental mode, where the usual constraints are loosened and ideas can combine in new and unexpected ways. But perhaps the most remarkable discovery about sleep in recent years is a purely mechanical one. In 2012, a team led by Macon Nedergaard at the University of Rochester discovered a system in the brain that essentially acts as a waste removal mechanism, and it operates primarily during sleep. They called it the glymphatic system, and here's how it works. During wakefulness, the brain's cells are packed tightly together, leaving very little space between them. But during sleep, those cells shrink slightly, increasing the space between them by as much as 60%. And through those widened channels, cerebrospinal fluid washes through the brain, flushing out waste products that accumulated during the day. Among the waste products being cleared are toxic proteins, including beta amyloid, the protein that forms plaques in the brains of Alzheimer's patients. Think about what that means. Sleep is literally the brain's cleaning cycle. While you sleep, your brain is being washed, and if you don't sleep enough, that waste accumulates. Chronic sleep deprivation has been linked to increased risk of Alzheimer's disease, and one possible mechanism is that the brain simply doesn't get enough cleaning time to clear the toxic proteins. This discovery has changed how the medical community thinks about sleep. It's not optional maintenance, it's essential infrastructure. And right now, as you lie here, your brain is beginning to shift into the rhythms of sleep. The neurons in your brainstem are adjusting the balance of neurotransmitters, reducing the excitatory signals and amplifying the inhibitory ones. The reticular activating system, which keeps you alert during the day, is being quieted by sleep-promoting neurons in the ventrolateral preoptic area of the hypothalamus. There's a beautiful name for you. Your cortex is beginning to generate slower, more synchronized waves of electrical activity. Alpha waves giving way to theta waves, your consciousness gradually dimming like a light on a dimmer switch. And as it does, your brain will cycle through stages of sleep, each with its own characteristic patterns of neural activity. Light sleep first, then deeper slow-wave sleep, then REM sleep, and back again, in 90-minute cycles that repeat throughout the night. During slow-wave sleep, the hippocampus will replay what you learned today. During REM sleep, your brain will weave strange narratives from fragments of memory and imagination, and you may or may not remember them in the morning. And throughout it all, the glymphatic system will be washing your brain clean, clearing the metabolic debris of the day, preparing the tissue for tomorrow. Now, let me talk about one more thing, because it connects to everything we've discussed, and it's something that affects millions of people. The brain, for all its astonishing capabilities, is vulnerable. It's a physical organ, made of cells and chemicals and structures, and when those go wrong, the consequences can be devastating. Consider Alzheimer's disease. Neurons in the hippocampus and surrounding cortex begin to die. The brain literally shrinks. Because the hippocampus is one of the first regions affected, The earliest symptom is difficulty forming new memories, the same deficit that HM had, but progressing relentlessly, gradually erasing language, personality, the ability to recognize loved ones. Despite decades of research, we still can't halt or reverse the disease. Parkinson's disease destroys the neurons that produce dopamine in the substantia nigra. Without dopamine, the motor circuits can't function properly, producing tremor, stiffness, and slowness of movement. We understand the circuit remarkably well. We know which neurons are dying and which neurotransmitter is missing. We can temporarily replace it with L-DOPA. But the underlying degeneration continues. We're treating the symptom, not the cause. And depression, perhaps the most common brain disorder, turns out to be far more complex than the simple chemical imbalance story. SSRIs help many people, but they increase serotonin within hours, while therapeutic effects take weeks, suggesting something deeper is happening. Disruptions in connectivity, neuroplasticity, inflammation, and gene-environment interactions. The good news, and there is good news, is that the tools we've been talking about tonight are accelerating our understanding of these diseases faster than ever before. Optogenetics is revealing the specific circuits involved in depression and anxiety. Brain imaging is showing how different treatments alter brain activity. Genetic studies are identifying risk factors, and new approaches like deep brain stimulation, where tiny electrodes are implanted in specific brain regions and deliver precisely calibrated electrical pulses, are showing remarkable results for Parkinson's and are being explored for depression, OCD, and other conditions. We are, slowly, learning to speak the brain's language. And you know, lying here tonight, letting all of this settle over you, there's something deeply moving about the whole enterprise. The brain studying itself. Human beings using their brains to understand their brains. Building microscopes and scanners and electrodes, developing theories and running experiments, accumulating knowledge about this 3-pound organ that somehow contains everything we've ever experienced, everything we know, everything we are. Cajal, drawing neurons by candlelight. Hodgkin and Huxley, spending summers on the English coast, teasing apart squid axons. Lowy, stumbling out of bed at 3 in the morning with an idea he couldn't afford to lose. Candell, patiently studying sea slugs for decades, unraveling the molecular basis of memory. These people used their brains to understand brains, and the recursive beauty of that never gets old. And the universe they revealed is stranger and more wonderful than the ancient Egyptians or Aristotle could have imagined. The brain is not a cooling system. It's not a container for fluid spirits. It's the most complex structure we've ever encountered, a living network of billions of tiny electrochemical machines wired together in patterns of such intricacy that we've only begun to map them, generating thoughts and feelings and consciousness through mechanisms we're only beginning to understand. And right now, yours is doing something remarkable. It's winding down. The neural circuits responsible for wakefulness are gradually yielding to the circuits that promote sleep. Your breathing is slowing. Your muscles are relaxing. The stream of thoughts that has been carrying you through this story is beginning to fragment, to drift, to lose its linear coherence. And that's fine. That's what brains do when the day is done. Think about this. One last thing, if you're still here. Right now, trillions of synaptic connections throughout your brain are active. Some are strong, some are weak. And that specific pattern of strengths and weaknesses encodes everything you know, the name of your best friend from childhood, the taste of your favorite food, the words to a song you haven't heard in years. It encodes the skills you've learned, the places you've been, the faces you recognize, the language you speak. It even encodes whatever fragments of tonight's story you'll carry with you into tomorrow. Because listening to this, right now, has physically changed your brain. Some synapses have been strengthened. Some new associations have been formed. The brain you wake up with tomorrow will be, in some small but real way, different from the brain you had when you started listening tonight. That's what brains do. They change. They learn. They grow. And when you sleep tonight, your hippocampus will replay some of what you heard, sending echoes of Cajal and Hodgkin and Lowy through your cortex, strengthening the new connections, weaving new knowledge into the vast tapestry of your neural network. Your glymphatic system will wash your brain clean. Your dreams may weave strange stories from fragments of neurons and synapses and sea slugs. And in the morning, you'll wake up, and your brain will boot back up, resuming the extraordinary performance it has been giving since the day you were born, constructing a world, generating a self, navigating a life, all from the quiet firing of 86 billion neurons doing what they've been doing, in one form or another, for 500 million years of evolution. That's what animal brains do. That's what your brain does. And the fact that it can do all of that, and also wonder how it does all of that, well, that might be the most astonishing thing any brain has ever accomplished. So let it rest now. Let the neurons slow. Let the waves wash through. Let the ancient machinery of sleep take over, as it has for every animal with a brain, since long before there were words or thoughts or questions about any of it. The brain knows how to sleep. It's been doing it for a very, very long time. Trust it, and drift off.